Please welcome Alexandre Savio with his talk about Nipy and functional brain MRI. Welcome.
Okay, thanks for coming, everyone. Thanks to the organizers. So I'm going to talk about Nipy, which is a set of projects on brain image processing. So I'm Alexandre Savio. I have been working for some time in the Klinikum Hess der Isar, which is the hospital of the Technical University of Munich. You can find me as Alex Savio from, well, I heard I'm very Googleable. So this is the outline of the talk. I'm going to show you and explain a bit what are the Nipy modules. I'm going to show you some of the databases available online. Also, I'm going to talk about the format of the files, what is functional MRI, and a bit of the pre and post processing of these images, if I have time. So Nipy is a set of projects, as I told you. If you go to nipy.org, you can. Okay. So there are many projects related with brain MRI and also MEG and EEG. I'm going to explain you a bit of the ones I know. If this works. So the first one is Nibabel. It's a library to read many types of neuroimaging file formats. It's quite handy if you are dealing with different tools for brain image processing. So it's just a library to read and write brain images. Nipy is the origin of all this. It's a large set of functions and submodules for brain image processing. It has some machine learning stuff, registration, statistics, time series, visualization. But then there's more specific modules. For example, Nipy, which is a module that allows you to build workflows of image processing, and so it is like a wrapper that works as it's a wrapper around different softwares from different universities, and it offers you the same interface to interact with these tools. And you once you build your workflow, you can run them in batch or even in a cluster and parallelize the whole preprocessing of a big database. Then you have NeTime, which is for time series analysis for fMRI, functional neuroimaging. Then you have DPI, which is for diffusion image, which is to work with fiber tracts of the white matter. Then a very recent one is NeLearn, which is machine learning for brain image analysis. It uses scikit-learn to many typical processes on brain image. So I'm going to explain a bit what's functional MRI. Well, an MRI scanner is a very fancy equipment. It uses two magnetic fields, very strong, to spin the atoms of your body and detect the energy that they liberate when they go back from this spin, more or less. This is a very summary. So I'm going to talk about fMRI, which is basically measuring oxygen consumption and blood flow of the brain. So it's a signal known as BOLD. So as I told you, MRI is also functional MRI takes many volumes of the brain in time, and it acquires one slice, so slice per slice, and then it goes back. Okay, so I'm going to present some of the databases that are available online. First, well, we have in this page the Indie project. Here you have many different big databases of neuroimaging with different types of MRI. One very popular one is called ABIDE, which is with images of autism disease patients. There is, if you don't want to pre-process all these images, because actually it's a lot of work, there are some databases that you can acquire them, you can download them already pre-processed with a specific pipeline. Here in this webpage you can find some, and you can also use NiLearn. They have the datasets submodule where you can download, for example, a preprocessed ABIDE, a preprocessed ADHD, preprocessed COBRE, also OASIS and the New York University Resting State database. This is all very documented. If you go online on the NiLearn website, you can find better description of these datasets. So I'm going to show a bit what is the file format of these images. First, well, the extension is nii.jz when it's compressed, and just .nii when it's uncompressed. So I downloaded a subset of ABIDE too, and I'm going to show you a bit how is this database. So here I have the directory where I downloaded it. I have one directory for each subject. I have the session of the acquisition of the subject. Then I have the anatomical or structural image. And the rest is for resting state fMRI. So it's an fMRI image. Sorry. Okay. So I'm going to So okay, so I'm getting one of the subjects. I'm using a tool that I built for this kind of data sets. So here I have the path of one subject's rest fMRI and the path of the structural fMRI. I'm going to use Nibabel to load the image. I import this as nib.load the file. And if I get an object of type nibabel.nifty1.nifty image. So this file has metadata. It has a large header and also the file image. Oh, this thing. So if I use get data, I get a NumPy array, which is the actual image of the actual brain image. And if I want to check the shape of this data, so you can see it has 4 dimensions. The first 3 dimensions, they are spatial dimensions, so X, Y, and Z. Well, I don't know Okay. So X, Y, Z, and this is time. Okay, so I have 162 volumes of brain images, fMRI, and okay. Sorry. Okay. So this nifty image object has also a big header. It shows me things like, for example, I have the dimension of the image. I have the dimension of each pixel of the image. I can't scroll down. Well, yeah. So I can use the header as like this. So I can pick each field of the header and, for example, read the pixel dimension. It also has an affine transformation matrix, which converts the coordinate space of the pixels to physical space in millimeters. I can also get the number of slices that the image has. So depending on what direction the image has been acquired, it tells me how many slices it has for each volume. Well, you can check better in Nipy how the nifty image, so how the nifty file is and how to work with it. I'm going to use an iLearn to plot these images. So, well, it has a plotting submodule. I'm calling it NiPlot. Also an image, which is an image submodule that I am calling NiImage. And, well, I'm taking the third volume of this fMRI image to plot, just to show you 3 orthogonal slices of one of the volumes in this fMRI image. Of course, it will be much nicer if I could show you the whole temporal series. So, well, I tried. This is not going to fit, but yeah, but I have to get out of here, and it's not allowing me to. Okay, thank you. So, well, I made a video with Matplotlib, and so these are the volumes of the fMRI, of the raw fMRI image. Well, I had one thing is, well, to plot it correctly, I had to set the contrast of the image, like with these parameters Vmin and Vmax, and so here I'm using NiLearn and Matplotlib to create this video. Okay, so I'm going to talk a bit about the standard preprocessing steps of the fMRI data. The first thing is to remove the first 6 seconds of the fMRI data, because the magnetic resonance field takes some time to stabilize, so the first images are usually bad. So I define a function to trim this image, and okay. What? Well, and it's going to calculate how many volumes I have to remove of the data, and instead of having 162 volumes, I have 159. Um, Slice timing and motion correction. Well, slice timing is about so this is a temporal series. So each volume should be in the same moment, but the problem is that the acquisition has been taken. So one slice in different moments. So what we have to do is to interpolate each slice with the next ones to have the whole volume more or less. So we invent data, but we are interpolating to make each volume in the same time. I don't know if that's clear. So here I have the slices. So the first slice, second slice, third slice, and they all have been taken in different moments. But I need the whole volume in the same moment of time. So for that, I need to understand how this image was acquired. I need to know what model of the MRI scanner. I need to understand the repetition time, which is the time that takes to acquire one whole volume, so all the slices. How many slices were taken? What is the orientation? So axial means from the head to the feet. In what order? Ascend, which means it was taken from the feet to the head. And it seems that for this Philips scanner, I needed to know how many packages they used for the acquisition. So I Googled it, and I found some explanations from my university on what is the order of these slices. Yes. So I used a Nipy. They have a sub module called time funcs, and there is a function that I give to it the number of slices and the repetition time, and it will give to me a vector of 45 numbers, which are the seconds, the relative seconds that each slice has been taken for me. Yeah. So the first one is the 0 here, and the second was 0.055 seconds later, and so on. I'm going to apply this interpolation process. Thankfully, Nipy has another function which is called space-time realign, where I give the image, the repetition time, the slice times that I obtained. Yeah. So, well, I'm going to cache it because it takes a lot of time to run this. And, well, I actually ran it. And I'm going to show you a bit of the... So, there's another problem that space-time realign is doing. It's not only about the times of the slices, but also the subject might have moved during the whole acquisition. So it's also correcting for motion, and well, it's not about only head motion. Even when you are acquiring a subject, there is a lot of respiration and cardiac motion that this could correct for a bit. So this function does both things for me. Slice time correction and motion correction. So I'm going to show you this, which is the estimated motion during the whole acquisition. So after running the space-time realign, it gives me the translations of each volume relative to the first one. So the first one starts in 0, and this is motion estimated across the functional MRI. So these are the 160 slices. Sorry, 160 volumes. Well, this is in millimeters, and well, it's in fact this is not too much motion. 0.05 millimeters, 0.1 millimeter is not too much. So the image is quite good. So well, I'm showing you the shape of the image is still the same as before. As I have 160 volumes, the same size. The thing is that the Nipy also has an internal type system of images. So in fact, what it returned to me was a Nipy image, and I really don't. I prefer working with the Nibabel things. So for now, my only solution was to save it on hard drive and load it with Nibabel. So the well, realigner. So the space-time realign also gives me an affine transformation to apply to the image to correct. So this is what. So, well, in fact, I have one affine matrix for each volume of the fMRI image, and I can see all of them to see how much, so exactly how it transformed each of the volumes. And then I created a video of the realigned image. I think you won't see too much difference here, but well, thankfully the image is quite good. Well, so this is rest in state fMRI. What does that mean? It means that we are not interested in fast changes in the signal of the brain. We are interested in the signals related with rest in state activity. So so we are interested in slow changes. So one typical thing to do is to filter, to do a temporal signal filtering on these images, and there is a band very known in biology that we know that it's about rest in state, and this is the band from 0.1 hertz to 0.01 hertz. So well, I copied this function from somewhere else. I improved it a bit, and so this is I give it the file, the low pass frequency threshold, and the high pass frequency threshold. It's not complete here, but because well, the size of the cell is not good. I apply it to my realigned image. I save it, and I create the video. Well, here you cannot see very well. I still don't know why the video is not good, but I can show it to you with another tool that I have here, FSL View. Well, the contrast is not very well here. The contrast in the projector is not very good to see it, but well. So what I'm doing here is filtering the functional signal to get only signals in the band, the time band that I wanted. So after this, we are going to smooth it because there's a lot of noise in this image, and after the whole processing that we did on it, we need to correct it for all these processes. So I'm going to use from Nylearn. There is a function called smooth image, where I give my filtered image and the size of the Gaussian kernel that I'm going to use to smooth it. So So FWHM is the full width half maximum of this kernel in millimeters. So I'm going to use a 80 millimeters Gaussian kernel to smooth it, and I'm going to store it and show you the video again. But yeah, this one has the same problem as before. I still don't know why. And well, so this is smooth resting state signal from the brain image. I know you cannot see too much here, but there's a lot of people studying the patterns in these waves. Okay, so one interesting part to reduce the amount of computation we have to do on this kind of images to calculate where the brain pixels are. So where I'm only interested in the brain. So why should I apply some signal filtering or other things to the image? So Nylern has a masking submodule and a compute API mask function that will give me a mask. So is a volume, one 3D volume, showing what with ones and zeros where is the brain in this image. So as you can see, the shape is the 3D part of my fMRI image, and it only has zeros and ones. I'm using the NumPy unique function to get what values the mask has, and it's only zeros and ones, just to show you. Yeah. Well, this doesn't seem to make much sense, all this. Well, but this is just to show you the mask over the fMRI image. So I'm using I'm using index image, and on my realigned image, I'm getting the first file, so the first volume. I'm plotting it, and I'm adding the mask as edges, and it will show me the mask over the fMRI just to check if it's more or less correct. It's not very good, but for some things it works. So apart from the rest fMRI data, in ABIDE I had also the anatomical image. Is also called structure or T1 weighted. This image gives you the best resolution, so it has a very high resolution, and gives you the best contrast of the tissues of the brain. So plotting it, you can see the image. This is a 3D volume. Sorry. And with this image, there are some processes to segment it to get the gray matter of the brain, the white matter. For example, so here I'm using Nipy. This interfaces submodule, I'm using SPM, which is a tool in MATLAB, but I'm running it from Python. And I'm using the function new segment a new segment is going to segment the image into gray matter, white matter, and fluid. I didn't have time to run this, sorry. And well, there's a big problem. In fact, after my talk in NeuroPython, someone sent me an email, and what about doing connectivity or So there's one step which is called spatial normalization or registration. So all our brains are different, and we have to normalize them to the same space and put them to do the statistics. So in many university groups have tools for this. There's no tool in Python to do this yet. So you can use Nipy to run this. This is going to be my next talk. Yeah. So here I'm showing you how to use normalized 12 from SPM to do this registration. Okay. So this is finished. You can download the Docker file to have all these tools installed for you. And I'm open for questions, and my boss is looking for a PhD student to work on this, on Python. Thank you.
Thank you so much, Alexander. Do you have any question for him? Okay.
Hi.
Hey.
I was interested in the space-time algorithm you showed for the normalization. In particular, as far as I understood, you show that at some point the algorithm estimates what should be the movement, considering the images. So can you provide us more details on it?
Yes, there are many ways to do that. So you have different volumes, and basically you have to choose one volume of reference, and you apply a registration from one volume to this reference image. So to this reference volume, you apply registration for each of them, and then so this registration is going to transform your image to be similar to your reference image, and you apply this transformation to your image. Yeah, so this is one way of doing it. So in basic it's a linear transformation with an affine matrix. You can more or less solve this problem. It's true that there's more things involved, because you can get non-linear errors in the image because of magnetic field effects. But this is more complex to solve, and not many people do that. Yeah.
Any other questions? So I'll drop a question. What are the patterns that are you already exploring the data, and yeah, if there's any insights about the patterns, or if you're working on that?
Yeah, well, I have been working with Alzheimer's disease. Yeah, there are many publications saying that, for example, so the patterns in these waves, they show you areas of the brain that work together. So they're called rest in a state networks, and it's shown that for Alzheimer's disease patients, for example, these networks, they are affected. Yeah, and well, there's a lot of research on that. And in fact, Daria, my friend, she's going to talk a bit more about the post-processing of these things.
Great, so you can follow also Daria talk.
Yes.
And there's another question here. Okay, so thank you so much, Alexander.
Thank you.
Okay.